Friends, today in this video we will discuss about the mechanism of RNA polyadenylation, and this RNA polyadenylation is the addition of this poly-A tail to the 3' end of RNA. So in my previous video I have discussed about this capping process, the 5' capping process. So this capping process as well as the addition of poly-A tail and the removal of intron, which is a splicing process, these three processes are very important for the formation of mature and functional RNA. So the first event that takes place for the formation of this mature RNA is 5' capping, followed by the splicing that involves the removal of intron from this coding sequence, and then the addition of this poly-A tail. So in this video I will discuss about the mechanism of how this poly-A tail is added to the 3' end of RNA. So the proteins that are involved in this polyadenylation process are cleavage factor 1 and 2, okay, the polyadenylate polymerase, and the cleavage stimulation factor, and the cleavage and polyadenylation stimulation factors. So all these factors plays a very important role in this polyadenylation process. So what happens in this RNA, there is a sequence, this AAUAA sequence is present that directs the recruitment of this CPSF protein. So this CPSF protein is recruited at this site, and the downstream of this AAU site there are two sites. One is the cleavage site where the cleavage will occur, and another one is this GU rich sequence where this CSTF protein will come and bind. So in the 3' end of RNA there is two different sites are present that will dictate the recruitment of these two different factors, the CPSF, which binds at this AAUAA sequence, and this CSTF, which will go and bind to this GU or U rich sequence. So once they sit in this sequence, then these three factors come and perform their respective function for the formation, for the addition of this poly-A tail. Okay, so how this process occur, let us discuss in this slide. So as I've said you that once this RNA polymerase reaches this AAUA site, the CPSF and CSTF comes and binds to their respective sites. So the CPSF as well as the CSTF, they are already present from the assembly of these transcription factors before the initiation process. That means that both the CPSF as well as the CSTF are present in this RNA polymerase C-terminal domain. Okay, so these two are present, and once they encounter their site, they move from RNA polymerase to their respective sites, and the binding of the CPSF to this AAUAA region is very weak, as well as the binding of the CSTF to this GU rich sequence is also very weak. So the interaction between these two protein, this subunit 160K of CPSF as well as the subunit of CSTF, the 77 kilodalton protein, both of this subunit stabilizes their interaction just because their interaction is weak. So CSTF 77K domain stabilizes this 160K domain of CPSF, and this stabilization helps them to remain adhered to their site. So once they get recruited, the CF1 and CF2, these cleavage factors, come, and at the downstream of this AAUS sequence is the CA site where this cleavage will occur, followed by the addition of poly-A tail. So what happens once they get recruited, cleavage factor 1 and cleavage factor 2 cleave this site, and this PAP protein will start adding the poly-A tail. Now the addition of this poly-A tail at this cleavage site is very slow. Okay, one more event happens is that after the cleavage, the CF1 and CF2 gets detached from this RNA. However, all of these protein complex are present. Okay, so this addition of A tail by PAP is very slow, and its addition is further accelerated by the recruitment of this PAB2 protein. So PAB stabilizes this PAP protein, as well as it also increases the processivity of this PAP protein. So the rate of addition of this poly-A tail increased by the recruitment of this PAP protein, and it also stabilizes this PAP protein. So after all this event, the poly-A tail is formed. Okay, and this is how this proteins plays an important part in the addition of poly-A tail. So CPSF and CSTF, which were present in this RNA polymerase, they jumps to the RNA when they encounter their respective sites, such as AU rich region as well as GU rich region. And once they come to their site, PAP, CF1 and CF2, all this protein gets recruited. PAP can interact with the 160 subunit of the CPSF, and this cleavage factor cleaves, and after its cleavage they gets removed from their site, and PAP start catalyzing the addition of this poly-A tail. Addition of poly-A tail is very slow. So PAB2 protein comes, it serves two different functions. One, it increases the rate at which this poly-A tail will added, and the second one is it stabilizes this poly-A protein. This poly. So it was previously reported that RNA polymerase, after reaching this AU rich site, the polymerase pauses at this site and gets detached from the mRNA, thereby leading to termination. While certain factors says that polymerase 2, after reaching this AU rich region, the several protein gets removed from the C-terminal domain, which leads to the removal of this RNA polymerase, followed by its termination. So different theories has been proposed for this termination process. And one more thing that I would like to say, that in this case, no phosphorylation of CTD is necessary, as I have discussed in capping that C-terminal domain needs to get phosphorylated for capping process to happen. But in this case, there is no requirement of the phosphorylation of CTD domain. So guys, this is the process by which polyadenylation takes place, and in case if you find any difficulty, you can put your question in the comment section. Thank you.